When I landed in Antarctica, I was hooked. It was like being an astronaut on another planet, but we were here on Earth. And I decided I would dedicate my research to becoming a polar scientist.
When I had the opportunity to go to Antarctica, I took it and I went there and I said, oh, this is my place. So since then, I have been working in polar regions.
Seeing a humpback whale breach for the first time, seeing a pack of orcas 30 strong cruising through the Antarctic environment at what seems like effortless speed. It's a miraculous experience, to say the very least.
Welcome to another episode of Behind the Science. My name is Damon Stanwell-Smith and I'm the Head of Science and Sustainability here at Viking. And today I am at the beautiful town of La Jolla in Southern California at the Scripps Institution of Oceanography and the J. Craig Venter Institute, where I'll be learning more about the genomics work that is being undertaken by the scientists here based in California and also on the expedition vessels, where we are collaborating on developing cutting-edge ways to undertake marine research and monitoring in remote locations.
My name is Professor Andrew Allen, and I am a joint professor between the J. Craig Venter Institute and the Scripps Institution of Oceanography. Our lab is physically located at the J. Craig Venter Institute, but it's really a collaboration between JCVI and UCSD, which is where the Scripps Institute of Oceanography is. And altogether, we have a lab of around 20 or 25 people, maybe 10 or 12 PhD students. And in our lab, we work on the diversity and biochemistry and metabolism of photosynthetic organisms in the global ocean. And we've been working in the Southern Ocean in and around Antarctica since around 2010. The Southern Ocean is known as a high nutrient, low chlorophyll ecosystem, which means it has high concentrations of nitrogen in the sunlit layer that do not get completely consumed. And this was a mystery for many, many decades. But it turns out that the reason that these high nitrogen levels don't get completely consumed is because there's iron limitation. And when there is iron supply from dust or volcanoes, it enables nitrogen to be consumed. But when the nutrients and phytoplankton in the surface layer of the Southern Ocean are subducted and remineralized to depth, they provide the nutrients that are upwelled into temperate ecosystems. So what happens with drawdown of micronutrients and nutrients in the Southern Ocean has a disproportionately important impact on upwelling of nutrients, what types of nutrients and the ratio in which they occur in the California current upwelling ecosystem or the upwelling ecosystem along the coast of Chile and Ecuador and Peru. And it's also warming quickly, warming faster than other parts of the ocean. So we're particularly interested in the interplay between increased carbon dioxide and temperature and these nutrient uptake mechanisms. And the Southern Ocean is home to extraordinarily beautiful populations of diatoms and other phytoplankton, large phytoplankton. So if you're interested in photosynthetic plankton, the Southern Ocean is a really exciting place to look under a microscope. But what is becoming clear is that we need a much, much higher level of sample collection and sample throughput to maximize the actual utility and use of this type of technology. So I think that our monitoring approaches to date are a bit prehistoric almost, very, very rudimentary. We're in the very, very early days of figuring out how to do this properly. And what we're really missing is consistent access in the ocean, almost all the time. But sampling one or two locations once every 3 or 4 years, which is classically how the Southern Ocean has been sampled, is just really insufficient and inadequate to piece together how an ecosystem like the Southern Ocean is responding to modern environmental perturbation.
My name is Maria Vernet. I am a research biologist at Scripps Institution of Oceanography. The research that we do in my lab is on phytoplankton, mostly on the ecology, meaning how do these cells that have been around for millions of years relate to the environment, and also what do they have in themselves, what is their ability to withstand these challenging environments. One of the limitations that we have, it takes months for samples to come from Antarctica. It's very expensive to bring them. They have to be frozen. How do you freeze things at sea? How do you freeze things in the plains? How do you freeze things when they are stuck in customs? So the more that we can do at sea, it makes us approach the environment, and by being there and seeing how the environment works, it really gets the juices going. We think about it. What else we can do? What else can we measure? How else we can learn about the environment now? Not when we are here. Otherwise, we come home, we write a proposal, it takes years to get it funded, and then we get the idea, and then we go to sea. By the time I wouldn't say the question is old, but we could have done it much faster. And we are in a time in which the polar regions are essential for us. If the polar caps melt and we cannot convince people that we need to preserve them, then we are going to be in a wealth of trouble. And we are leaving that to the next generations. And we as scientists are very conscious that we need to share our knowledge and our findings as fast as possible.
My name is Alison Cusick, and I am a biological oceanographer at the Scripps Institution of Oceanography and the J. Craig Venter Institute. I'm currently in my final year of my PhD, working on my dissertation where I'm looking at how melting glaciers influence the base of the food web, or which species of phytoplankton are living in the coastal region on the Antarctic Peninsula. My first time to Antarctica was in 2013, so that now makes it 10 years since I first went into the realm of polar research. I have spent, over the last 7 years, over 300 days on expedition vessels in Antarctica.
Well, one thing that we'd like to understand in particular is how temperature is impacting nutrient uptake mechanisms. So there's a lot of interest in understanding the ways in which iron availability regulates climate on glacier and interglacial cycles in the Southern Ocean, and there's been a lot of mystery associated with why iron is not used even more efficiently than it should be. And we've come to understand that it has to do with carbonate concentrations. So we're increasingly interested in understanding the details around iron, carbonate, temperature interactions in the Southern Ocean. So we really need to understand what are the molecular and genetic mechanisms that govern phytoplankton responses to temperature in the Southern Ocean.
Fjord Phyto is a program that works in collaboration with tour expedition vessels visiting the Antarctic Peninsula, and travelers on vacation can help collect scientific samples that are then sent back to me and my colleagues in the lab, where we process these samples to understand what was in the water at that time and how the season might be changing. So the program is called Fjord Phyto, and we're looking at specifically phytoplankton in the fjords in the coastal region. And this program was designed in 2016, and all of the data that comes from it has contributed to my PhD dissertation work. We first brought Fjord Phyto on board the Viking expedition vessels in March 2022, and that's also when my colleague Martina and myself were invited on board. During that time in Antarctica, we filmed a Viking.TV special episode on the research that was happening during that month. Once we're out in the water, we sample plankton. Two different methods. One is using a phytoplankton net, and the other is just taking a sample of seawater. And the seawater sample can give us a quantitative measure of how many phytoplankton are in that volume of water, as well as the species identification. And then the net is how we concentrate enough biomass that we can preserve that sample to later do DNA sequencing back in the lab.
My name is Martina Massioni. I'm a doctor in natural sciences. I'm a biologist, and I work in the National University of La Plata in Argentina. I did my PhD with most of the samples from Fjord Phyto. Fjord Phyto has been a long journey, actually. I've been there since the beginning, since we started back then in 2016. The key findings for Pure Phyto was like when we started looking at the samples and we went back to the literature and we say, okay, how is this changing, or how is it always been the same or not? And we realized like most of the places that we sampled with Pure Phyto haven't been sampled before, at least for phytoplankton. So we're kind of starting to understand these different areas like the fjords and like how these embayments or these areas, or sometimes there are like little islands that nobody had ever sampled, and see what is the phytoplankton there, how do they look like. But also like what I did at the end of my PhD is like try to see the 4 years that I have samples for and try to understand how the community was changing through the season. And I kind of put together like, okay, this is what happens at the end of the season, at the beginning of the season. This is what I should find in the sample, and this is what happened in the middle of the season, and this is what happened at the end of the season. So trying to hypothesize how the community is changing through the season and also through the years.
In the Viking episode we filmed in March 2022, you had asked Daniel and myself a question of what do we see as the future. One of the incredibly cool things that came out of the pandemic were these genomics PCR labs that the Viking ships had on board, and we had said that it would be amazing if those could be converted to doing research or genetics at sea. So while the ships are traveling, we can in real time start to understand something about the communities of life that live in the water and start to really track immediately. Instead of preserving a sample for an analysis later down the road, we could do it right on the ship. And so it's awesome to see that 2 years later that is being carried forth.
But with genomics at sea, it's like if we can see that on the field and we can take a sample and look at the microscope and see what is in there and see the sequence right away. And so we're like, oh, this is interesting. Oh, maybe this is the new species that Martina found, or like somebody else look. And so we can also take a new sample and culture that, which is really important for describing new species.
We're trying something really unique and new with the Genomics at Sea program, which is of course sequencing DNA in real time in the field, which we are incredibly excited about. We think this is really a new frontier for marine science in general, to obtain DNA sequencing information from phytoplankton blooms and phytoplankton populations, as we pass through them.
I'm a 25-year-old, recently a 25-year-old PhD student here at the Scripps Institution of Oceanography. I also do a couple more jobs. I'm also a researcher on the Viking Octantis, and I've also been on the Viking Polaris in the past. So I am the lead scientist on the Genomics at Sea project, or GASP. It's a project that is involved with doing actual genomic analysis and sequencing while in the field. In the Antarctic environment, and the whole sort of idea behind GASP alludes to the importance of the Antarctic community and phytoplankton community in the Southern Ocean. That is very, very important to the actual induction of carbon dioxide and then production of oxygen. Some estimates give around 50% of oxygen production, CO2 removal. Those estimates tend to be a little bit more reserved when you look at more accurate measurements. We're looking anywhere closer to 70 to 80% of oxygen being produced directly by phytoplankton annually.
I first met Christian Johnson when he was an undergraduate student taking a class in spring. This was just after the pandemic and everything had been converted to Zoom online meetings. I was the teaching assistant of that class. And I ended up having Christian in 2 different classes, and he wanted to apply for one of the Scripps undergrad research opportunity programs, an REU. And so during the summer of 2021, Christian worked in the Vernay lab with me, helping to process fjord phyto samples. And since then, he was hooked on polar science. And after he completed his bachelor's degree, he started working in the Allen lab, continuing to work on fjord phyto and projects in the lab. And then when the opportunity came up to come down on one of the Viking vessels to do fjord phyto sampling and start developing ideas of his own, that is how this whole endeavor got started.
we had to design a portable genomics lab that we would be able to transport from here in San Diego down to Ushuaia to board on the ships before we can basically make the journey across the Drake's Passage and start doing our research. So that was definitely a very, very long list of not only hardware but also equipment and consumables that we would need to be able to basically do our entire genomics research project for the entirety of the season, spanning from around November all the way until March. It entailed, I believe, 5 different suitcases that all needed to be transported via ship to shore, which basically picks up at our doorstep, drops off at the port down in Ushuaia. And that was just the process of actually getting all of the gear down there. Before that, we had to do basically all of the laboratory preparation to know what exactly we were going to need for the entirety of the season, purchase all of the equipment that would be necessary. A lot of these different companies have back orders on everything, so we needed to make sure that we purchased everything well within advance so that it wasn't going to be one small piece of equipment that was going to hold back our entire first season of discovery down there. So once we basically had our list and then we were able to get it all transported down to Ushuaia, and then we had to get it loaded onto the actual ship, which was probably the easiest process of this entire long process that we're going through. And then once we had everything on the ship, there's a lot of different checks and balances that go into place to making sure that everybody is okay with space allocation, but also with preparing the actual laboratory space, because we were using the previous PCR space that they were using to do PCR analysis on the actual vessel throughout all of COVID to make sure that they were safe and secure throughout that entire time. So we had to basically clean out the entire laboratory and then set it up with all of our necessary equipment. There are another set of hurdles that are basically thrown at you when you're trying to go from the voltages that we use here in the US to the voltages that are used on Norwegian ships. So we had to have our entire electrical team gut out the entire laboratory, build in completely new electrical so that we would be able to use the equipment that we actually brought down there to be able to do our genomic analysis. And then once we finally got everything actually sort of situated within that room, it was just the sterilization and then actually just outfitting the very limited space that was in there with all of the necessary equipment and research devices to basically be able to do exactly what we were trying to do over the course of that season, the genomic analysis process.
The field of genetics or genomics is full of jargon, and the way I like to think of it is genomics is sort of this umbrella term. The word omics can kind of cover different ways of looking at genetics. So if you extract DNA, you can look at genomics through the DNA or the blueprint of an organism or any living thing. If you extract RNA, you can look at the gene expression levels or the genetic behaviors of different organisms, and that's called metatranscriptomics. And then you can look at maybe the functions within an organism's cell, which uses proteins. Every living thing has proteins that are doing the work within a cell, and that is proteomics. And then there's metabolomics. And now there's this newer kind of term eDNA that's catching on, and the E just means environmental DNA. So it's any organism living sheds DNA. This is where we're familiar with it in forensics or crime scene detectives picking up eDNA. In the environment, things are shedding DNA all the time. So eDNA is a way of sampling the environment, either sampling the air or in our case sampling the water, the ocean, to be able to look at what DNA might be left behind, and that can tell us something about the organisms living there or that had passed through that body of water, whether they're currently there or not. And that can be really powerful, of course, for understanding the microbial life, but also for big animals. Maybe you don't see a whale or you don't have a permit to sample a whale, but the eDNA might say there was a whale in the area from shedding whale DNA. This is a really cool way now to be monitoring the environment and life in the environment through genetics and eDNA.
So we have been involved with developing pipelines that allow us to do genomic analysis while in the field, particularly pertaining to how penguin colonies end up influencing the phytoplankton communities that are living around them.
Among other things, we would like to understand how eutrophication and nutrient inputs from penguin rookeries is affecting the type of phytoplankton in the water column and their metabolic activity and the types of molecular mechanisms they're using to acquire these nutrients. In addition to interactions between phytoplankton and bacteria. There are around 1 million bacteria per milliliter of seawater, and we think in the Southern Ocean in particular, bacteria and phytoplankton have undiscovered symbioses centered primarily around exchange of nutrients and vitamins and iron. And so we think that nutrients and organic matter from the penguin rookeries might affect these interactions. So we're interested in obtaining more insights into this.
In this past field season, we were using a sequencing platform called Oxford Nanopore Technologies. So we want to be able to validate the results that we produced, the novel results we were able to produce in the field with tried and true results that we produce in the laboratory, and we use Illumina sequencing here at JCVI.
In contrast to working in the lab in California, indoors, in very comfortable working conditions compared to working at sea in the Southern Ocean, well, it just could not be more night and day different. You know, in our ordinary laboratory environment, we have vendors that we can order things from, and they come the next day or in a couple of days. You know, if we're going to conduct research in the Southern Ocean, it has to be planned a couple of years in advance. Supplies and equipment all have to be in place, and we have to be very, very specific ahead of time about what our scientific goals are, what types of reagents and resources that we need. And now we're actually conducting genome sequencing experiments at sea to ask questions about variation in genomes within and between different populations of major phytoplankton.
When we're actually sequencing the genetic material, we're trying to break down the identification of the organisms. So in DNA, you're going to have what's called base pairs. You're going to have A's going with T's, G's going with C's. So what the nanopore, our sequencing device, is going to be doing is it's going to be pulling these pieces of DNA through small pores in the actual sequencing platform, and it's going to be real-time base calling, so basically reading back those individual parts of the DNA to us, and based on the actual sequence and the order that they're in, we're able to derive basically the identification of the organism from that. So that's going to be the actual sequencing portion of the whole analysis pipeline.
There really is no typical day, except for the Plan A agenda involves waking up early, having an excursion off the ship. One of those excursion options could be doing a science boat and sampling with Fjord Phyto, or walk around the penguins, or snowshoeing, kayaking, a number of activities you could do in the morning. You come back onto the ship to transition, have a meal, maybe take a polar nap, and then have an afternoon excursion where you go back out to do an activity or operation, and that could involve another science sampling boat. Maybe when you're back on board, you could host workshops or ways to get people involved who didn't go on that excursion. Every evening, there's usually a recap of what had happened that day. Maybe there's a lecture where you could talk more in depth about my specialty or something that you had heard people having conversations about that you want to go deeper in or share with people on board. And then you have dinner and a long day ends and you start it again the next day.
A lot of the people that are already on our team are very scientifically and technically sound. So we're not necessarily working on their understanding of the science or their understanding of the region as much as their science communication skills. So basically being able to work with the actual Viking guests, be able to communicate the research that's going on down there in a very broad and not quite as in-depth route as we usually do when we're communicating to the scientific community, and being able to help them to basically present all of the science in a much more relatable fashion to the guests that are on board, the expedition guests that are on board the Viking Octantis.
When we talk about using markers in genomics work or with DNA, you can extract DNA from a sample or your organisms, and you can choose different markers. They're like little barcodes that tell you a certain section of DNA that may be conserved throughout a certain lineage of animals or plants or protists, something you're interested in looking at. And that marker is sort of a placeholder so that when you sequence it, you get more information about that strand of the DNA, you can then compare that to these online databases where it matches it up to a taxa or species in the database. One of the challenges of doing this work at this current time is that there's no standard marker set to use. So many researchers use different markers depending on what they're interested in answering for their question. So if you're interested, just for example, if you're interested in bacteria, you may choose the 16S marker. If you're interested in phytoplankton, you might choose the 18S marker, and so on. You can choose, you can even customize ones that you are particularly interested in. Now, this gets tricky because a lot of the databases aren't well represented with species from all over the world, or particularly polar regions. So when you match these markers up to these databases, you might get incorrectly identified species. Now, this is where, the combination of Fjord Phyto and being able to sample with a vast number of vessels, but interfaces with the Genomics at Sea program, because in real time you can sequence what water you're sampling, and then with the Fjord Phyto samples later we can compare what my colleague Martina is analyzing under the microscope with what we're analyzing from the genetic markers, and then see how that's comparing in the databases. We're informing different methods for understanding phytoplankton.
At the end of the season, we collect all the samples coming from the different vessels that collect for Fjord Phyto, and some of those samples, the one that we put Lugal on, those are the ones that I analyze under the microscope. So what I do with the samples is I put a certain amount of the sample that I know of under this sedimentation chamber, so I can tell that everything is on that water is going to settle, and that settles overnight, and the next day I count everything I see under the microscope. So I have a specific count of what the species were there and how many of each one under the microscope. I mean, we focus on phytoplankton, but of course we found other things like zooplankton, diatoms, dinoflagellates, cryptophytes, or other flagellates that are very small. When I did my PhD, I was looking under the microscope and I was seeing all these things that didn't fit exactly what was described in the literature. So I was like, well, this seems like it could be like new species or new things for science, of course. And then that's when I realized like we need to merge the genetics and the microscopy together, because to describe these new species, new organisms that we found, we need both like a description of how they look like that the microscope can give us, like how they look, how is their shape, size, etc. And then the genomics is very important because it gives us like how is it the material, what genes are there. And so we merge those two and we can actually describe these new species.
With the Genetics at Sea program, if it's real time and you see an organism that you haven't seen before, or you can extract the genetic information or the RNA that tells you some behavior the cells might be experiencing, you can then look at your environment and maybe collect additional samples and information from what was happening that day. If you're just filling out a paper data sheet and you didn't think that you would need to care about how salty the water was, or if it was a windy day or a sunny day, you would just come back to the lab and only have the information that was written on that data sheet. Whereas now if you're in the field, on the ship, collecting a sample, immediately processing it to see something interesting, you could then know that all of that other extra information is right there. You can collect as well that same day.
One of the things that I can definitely say is the day that we finally ended up getting the sort of whole process to work, all the way from collection through DNA extraction and then onto the sequencer, and we saw results in the sequencer, was definitely the highlight of the trip. I know that there was a lot that had went into the actual preparation before and setting up the laboratory and everything, but if I had to say handed the keys over to my counterparts and my colleagues and we hadn't quite gotten the genomic sequencing up and running, not to say that it would have felt like a failure, but it would have felt like a failure.
what advice did we give Christian when he was embarking on this challenge of a lifetime to set up the PCR lab on the Viking vessel at sea? We wished Christian all of the luck in the world. We told him to try to just keep moving forward, not get hung up on the small problems that are certainly going to occur almost every day, and just try to focus on the things that are working, and make sure you're taking good notes about how we can do it better next year.
With any avenue of science, sometimes things just don't work, and that can be very, very frustrating. Even more frustrating when you lose a valuable piece of equipment that you need to be able to do your research. And since you're in such a remote environment, you don't really have access to any ports in or out for a very long time, you can't necessarily replace those. And if one of those does end up going into the water and not coming back out of the water, for example, a CTD that we lost in my first season when we were down there, that's definitely something that can put a little bit of a damper on the day. Especially when it ends up happening from something as seamlessly avoidable as a prop going through a line that is holding your CTD because somebody wasn't necessarily as diligent as they should have been with the retrieval of the device. But you, at the end of the day, have to realize that everybody makes mistakes. That's still a part of your team. Can't necessarily, especially when you're in such a remote environment and you are a part of a very small team, a small community. Can't let those things get under your skin. So it's figuring out what the next course of action was, and for us that season it was using the ferry box to be able to give us our physical parameters of the water column rather than having something that can give us a direct profile of the water mass that we were directly above. But it's sort of just rolling with the punches and realizing that, especially when you're in an environment that is so dynamic and likely to change so quickly, that things are going to end up coming up, and you got to be able to roll with the punches. We usually start operations right around, depending on the site, anywhere between like 5 and, if we're lucky, 7 a.m. So we're getting up early. We're basically getting completely dressed in all of our outdoor exposure gear to make sure we're protected from the environment. For myself and the rest of the team that are involved in these scientific projects, we then basically prepare all of the gear that we're going to need to take apart in the actual scientific collections while we're basically going to be going about for the entirety of the day. So we don't necessarily only have one project that'll be going on during that day. We usually have a multitude of different projects. So we will get all of the gear basically prepared and ready to be able to do all of the collections. And then we will go out, assess the actual environment first to make sure everything is safe to be able to be conducted properly. And after that field assessment is done, then we'll start actually doing the data collection for the projects that we have going on at that site that day. So far I just did my first year symposium, so we just got through with actually the first year of all of the coursework and everything, as well as the first field season in the Antarctic environment. I just did a presentation sort of wrapping all of that and trying to basically communicate that the best way possible.
We're just over the moon excited about this opportunity for Christian and for our lab and for the Scripps Institution of Oceanography in general. For Christian to conduct his PhD research in Antarctica. It's just an incredible opportunity, and I know that he's going to go on to do really exciting things, and this is really state-of-the-art training for Christian. To really understand what's going on with ocean phytoplankton responses to something like temperature, or carbon dioxide, or genomic variation, or genetic variability and biochemistry of uptake mechanisms, we need more people working on this problem. And I think the Southern Ocean in general remains critically understudied. One of the most exciting aspects of the collaboration with Viking is more consistent, reliable access to the Southern Ocean year after year. I've had numerous undergraduates and graduate students and postdocs ask me about potentially conducting research in the Southern Ocean now, because they understand what Christian is doing and what we have planned. And there's just an unending number of scientific questions, and we are particularly excited about training undergraduates and master's students and PhD students to have the skills to go into the field and address some of these questions. So we think that Christian is probably the first in what we hope are many students who will use this platform as an opportunity for thesis research and to conduct publication-worthy research.
We as scientists have learned that everyone is interested in science, not only the scientist. But the way of communicating science to non-scientists is different. So I think collaborating with the tourist ships and with Viking, I think is an excellent way to start doing that in real time. And having a monitoring in a difficult environment like Antarctica is extremely valuable and rich. The tourist ships that go there consistently for 4 months, and they can go to the same places and collect data, is really helpful, because we as scientists don't have the funding or the ability to sample to the degree in which for example, Viking can do it. So for me, we have other ways, maybe putting some moorings, but I have found that the icebergs break more than 50% of their moorings. That doesn't happen with the ships. So that is another way of why in the polar regions is so important. The other way why monitoring in polar regions is so important is because to define trends, meaning to define statistically significant changes in the environment, you need cycles, and the environment moves in cycles. You have ups and you have downs. In Antarctica, those cycles last for 10 years. And to define a trend, you need at least 3 cycles. We're talking about 30 years of collecting data. Nobody can do that as a single researcher. So partnering with people that go there for reasons of their own and they are interested in supporting the science is for us an excellent way of collaborating.
The Southern Ocean is a very, very difficult place to get to support research logistically. The funding for research in the Southern Ocean, despite its importance, is difficult to obtain. You know, historically, I think our scientific knowledge of Southern Ocean ecosystems in general, you know, lags behind by many decades of other regions of the world's ocean, just because scientists didn't get there until later, and scientific activity is limited by the extremely harsh logistics. So it's really difficult to work in the Southern Ocean. The National Science Foundation in the United States provides some access to the Southern Ocean. I think that there's widespread agreement in the scientific community that access to the Southern Ocean and to scientific resources there, should be increased. So our collaboration with Viking is something that we're particularly excited about. It's a really unique and valuable opportunity for us to get to the Southern Ocean more frequently, more regularly, to sample in more spatial detail. I think that what we're learning is that the ocean ecosystems are responding to change, but they're doing it in ways that are subtle and maybe a little bit more sensitive than we realized initially. And in order to see this type of changes that are occurring, we need more regular baseline sampling. And so interactions with Viking are providing this. It's a really unique approach. It's an interesting hybrid collaboration, and we are just really excited to be engaged in this collaboration.
I hope that you have enjoyed as much as I have getting to know more about the genomics work of the JCVI and Scripps Institution of Oceanography scientists, in particular the PhD work of Christian Johnson. We look forward to seeing him regularly on board our expedition vessels and hope that you too have the chance to share the excitement of such cutting-edge science as we go forward. When we set out planning the expedition ships to be cutting-edge oceanographic platforms to reimagine what ships of opportunity can do, it has been wonderful to see since we started in 2022 through to now in 2024 that new ideas can be brought to fruition and that new scientific endeavours, new ways to do field science, new ways to do monitoring have been possible. I look forward to seeing what's possible for the next few years and look forward to sharing that journey with you.